Welcome to our unit on genetics, which is one of my favorite in the biology course. So let's start with the basics of genetics. Heredity is the way organisms pass traits, which is a fancy word for characteristics, on to their offspring. And genetics is the study of how these traits are passed down through a population. So heredity is the passing down of the traits or characteristics, and genetics is the scientific study of how that occurs. In humans, eye color, skin color, hair color, height, and intelligence are some examples of genetic traits. Gregor Mendel is one of the first individuals to take a really detailed look at the field of genetics. He was an Austrian monk born in 1822, and he's widely known as the father of genetics. He bred pea plants to investigate how they inherited their traits, and he collected very detailed data, which is one of the reasons that his research was so revolutionary, because he collected piles and piles of data on different pea plant traits. And this was something that was necessary to really track how the traits were being passed through the generations. There was a lot of misunderstanding about how genetics was occurring at that time. So here are some of the traits that he looked at. He looked at seed form and color. He looked at flower color, pea pod color and form, and location of the flowers on the stems, and whether the pea plants were tall or short. He was fortunate in that pea plants have two major advantages as genetic specimens. First, they have many visible or easy to see traits, as you saw from the last slide. And secondly, they can be self-pollinated or cross-pollinated. Self-pollination is the movement of pollen from one flower, the male portion of one flower, onto the female portion of the same flower. So flowers, because they have distinct male and female parts, are able to pollinate themselves or reproduce using their own genes. And cross-pollination usually occurs with a pollinator, like a bee or a moth. And it can be random because those pollinators travel from flower to flower all over the place. And so the traits can be mixed from one flower to another, and it's hard to track how those traits actually occur because it's difficult to distinguish which parents were actually creating the offspring. So instead of using insects for pollination, which caused a random combination of flowers, Mendel brushed the pollen from one flower onto another so he could track the traits more carefully. So he was distinctly creating two different parent flowers and was then able to follow those traits more carefully through the generations. In order to keep track of the traits in each generation of plants, he created some terminology which we still use today. The first filial or F1 generation is the first generation of offspring created when two plants were cross-pollinated. So here are the two parent plants, and their offspring create the F1 generation. The second filial or F2 generation are the offspring of the F1 generation. So if these two plants were cross-pollinated, they would create an F2 generation of plants. And Mendel noticed that some plants always seem to produce the same trait in their offspring. He called these true breeding plants. So for example, if two purple flowered pea plants continued to produce purple flowered pea plants throughout future generations, he would have called that a true breeding purple pea plant. Other times, however, traits seem to disappear and then reappear in future generations. So sometimes when a green pea-podded plant and a yellow pea-podded plant were cross-pollinated, they created all green pea pods in the F1 generation, and then the yellow pea pods would show up again in the F2 generation. So they would disappear and then reappear. Mendel noticed that when two plants with different traits were crossed, the F1 generation only showed one of these traits, and the other seemed to disappear. When two of the F1 plants were then crossed, the F2 generation showed the trait from the F1 generation in 3/4 of the F2 plants, and the trait that had disappeared showed again in 1/4 of the F2 plants. So in this same picture that we just discussed, this yellow pea pod color disappears when two different plants were cross-pollinated. One of the traits disappears, and then in the F2 generation, 3/4 of the plants would show this same trait from the F1 generation, and 1/4 of the plants would show the original other trait that had disappeared. He was noticing that these proportions were happening regularly, and that's why his extensive data was helpful in following these traits and finding these percentages in each generation. What were Mendel's conclusions then? Mendel concluded that an individual's characteristics are caused by factors inherited from previous generations. And we now call these factors genes. A gene is simply a segment of DNA that codes for a specific trait. So if this is a chromosome, which we've discussed already, each of these bands on the chromosome is a specific gene. And each of those genes, in the simplest form, can code for a specific trait. It often does not work quite that simply, but for our purposes at the moment, each gene is coding for a different trait. And genes can have different forms called alleles. And considering that DNA hadn't even been discovered in Mendel's time, he really created some revolutionary rules based on just his pea plant observations. He also concluded that some alleles or forms of genes are dominant and some are recessive. This is known as the principle of dominance. So a dominant trait is the one that continued to appear in that F1 generation. It seemed to dominate the other trait. And the recessive trait is the one that disappeared in the F1 generation and then reappeared in the F2. So he named these dominant and recessive traits. In summary, Gregor Mendel used pea plants to learn about heredity. He suggested that traits were caused by factors inherited by previous generations, and some were dominant while others were recessive. Okay, so if these alleles are causing traits, then how are these alleles being passed down from one generation to the next to show the transfer of that trait through generations? In Mendel's time, the theory of blending was prominent. So this would be the theory that two parents' traits are going to combine in a blending way to create some sort of intermediate. So if you had a tall pea plant and a short pea plant, they might blend, and then the next generation would have a medium height pea plant, or all medium height pea plants. And Mendel's data, in fact, did not support that theory. The green and yellow pea alleles did not blend. Instead, in the F2 generation, the yellow allele seemed to separate from the green allele. What would you expect to happen to the colors of each generation of peas if the alleles were blending? So if we had blending occurring, if this theory was correct, and you were crossing a green pea-podded plant with a yellow pea-podded plant, we would expect that the next generation would be less distinctly green and yellow, that we would be getting more consistent greenish yellow pea pods as we move through the generations of pea plants. And that is not what Mendel's data was showing. Those yellow alleles were disappearing and then returning in full force. So combined with our knowledge of cell reproduction, which we just learned about, we now understand that alleles for a gene separate when cells divide. And because these alleles are passed down to offspring, this cell division must be occurring in the reproductive cells of an organism, which are called gametes. So mitosis occurs for processes like healing your skin, where an identical cell is being created to replace one, or that cells are dividing to create more identical cells. And that's happening in your body cells. But in order to pass genes down from one generation to the next, these changes need to be occurring in reproductive cells like sperm and eggs, or in pea plants in pollen and ovules. So here is the process known as meiosis, and we're going to explore the details of that a little bit later in this unit, but this is the process of dividing and creating more reproductive cells. Again, those are called gametes. And this causes Mendel's principle of segregation. So you can see that the two different types of genes, or two different alleles here, are actually being separated into different gametes. And he called that the principle of segregation. What are genotypes and phenotypes? So for a completely dominant trait, which we'll focus on for a minute here, each individual has 2 copies of each gene, one from each parent. So each new generation receives 2 copies of a gene, one from the mother, one from the father. And those genes, those alleles, can either be dominant or recessive. And the genotype is the genetic makeup of a gene, which we write in letter pairs. So I'll show you an example of that in just a second. The phenotype is the physical expression of a gene caused by the genotype. So a good way to remember this is phenotype and physical. So this would be a physical expression of a gene would be brown hair or blue eyes, tall height. Those are physically visible expressions of a gene. The genotype is not something that you can see. It's a representation. So let's take a look at genotype. Capital letters in a genotype represent dominant alleles, and lowercase letters represent recessive alleles. So here's an example. Dominant allele being the capital B. We usually name it for whatever trait we're discussing. So this might be a blue trait of some kind. We name that for the dominant allele, find the correct letter for that, and then use the same letter but in a lowercase format to show the recessive allele. So for example, the genotype below has 2 alleles, one dominant and one recessive. One allele came from each parent. So one parent donated a dominant allele and one parent donated a recessive allele. And the phenotype of this organism, so this is the genotype here, capital B, little b. The phenotype of this organism would show the dominant trait because the dominant allele overshadows the recessive. That's why we write it as a capital letter. And this is what Mendel was seeing when one trait was disappearing, was that one trait was overpowering, and that's the dominant trait. It was overpowering the recessive trait or recessive allele. So that recessive allele is going to disappear, at least temporarily, in some of those generations. So there are 3 possible genotypes for a completely dominant trait. So if we're talking about simple dominant and recessive traits, which we're going to get into more complicated traits soon, but if we're talking about completely dominant traits, in other words, there's a dominant allele and there's a recessive allele, there are 3 possible genotypes. 2 capital letters or 2 dominant alleles we call homozygous dominant. So both alleles are dominant. Homo means same. So these are the same, both dominant. Heterozygous is another option where one parent donated a dominant allele and one donated a recessive allele. Heterozygous. And then homozygous recessive means that both of the alleles that were received from the parents are recessive. And this is the only situation for a completely dominant trait in a homozygous recessive situation where you would actually see that recessive allele show up, because in both of these cases, the dominant alleles are going to overshadow the recessive. This one doesn't have a recessive allele. This one does have a recessive allele, but it's going to be overshadowed by that dominant allele, and so it's going to disappear. So if purple flowers are a dominant trait, any genotype with a dominant allele will be purple. Any genotype without a dominant allele will show the recessive trait, which is white. Both the homozygous dominant and heterozygous genotypes show a dominant phenotype. So let's try another one. If round seeds are dominant to wrinkled seeds, which are going to be recessive, what will the phenotypes be for the following genotypes? So here we're using R to represent the dominant trait of round. Capital R is going to be 2 dominant alleles, and therefore our phenotype should be round. Capital R, lowercase r, is also going to give a round phenotype because it has a dominant allele. And little r is going to be the one that shows the wrinkled recessive phenotype because it doesn't have that dominant round allele. So in summary, Mendel concluded that alleles are inherited separately within gametes. The alleles of the gamete are written with letters to form a genotype that shows as a phenotype. And again, these are, of course, are not actual letters in your body. This is just a model. A genotype is just a model that we use. So we represent those alleles with letters. So often when we're discussing genetics, the goal is to predict the outcome of the next generation. What is the next generation going to look like? And we use Punnett squares to solve those predictions. The principle of segregation, which we said was the separating of alleles in one generation to create the next, the principle of segregation can allow us to predict the probability or likelihood of alleles showing in future generations. So because those genes aren't just blending randomly, we can actually predict how these alleles and traits are going to show up in future generations. And one of the tools used to make this prediction is a Punnett square. Here's what a Punnett square looks like. So these 4 squares here represent offspring in the next generation, and these represent the genotypes of the parents. So it's a little bit like graphing in math, where you set up one parent's alleles on the top and one parent's alleles on the side, and then we are combining those alleles together. So this capital A is going to drop down into this box, and this capital A is going to come across into this box as well. So both of those parents, because these alleles are segregating in the gametes, they are going to distribute themselves independently from each other in the offspring. And so we can make some predictions about what these offspring are going to look like. Now, this does not represent 4 offspring necessarily. These are just proportions or the probability of these alleles being passed on. So we use these squares to represent percentages, where 1/4 or 25% of the offspring are going to be homozygous dominant, 50% of the offspring will be heterozygous, and 25% of the offspring will be homozygous recessive and show that recessive phenotype. So one parent's alleles are written on the top of a 4-square box, as I just said, and the other parent's alleles are written on the side. The same letter is chosen for both dominant and recessive alleles, as we've discussed, usually based on the dominant phenotype. And then alleles are combined to show the possible offspring combinations. Capital letters are written first for consistency, so that we don't have a little a and a big A over here that looks different than the capital A and the little a over here. This is the same phenotype here, same genotype, same phenotype. Both are heterozygous and are going to show the dominant trait. So in Mendel's pea plant experiment here, if we did a Punnett square for this, the dominant trait is going to be green. So if we are crossing a homozygous dominant green pea pod plant with a homozygous recessive yellow pea pod plant, the Punnett square is going to look like this. Capital G, lowercase g. So we've got one parent over here, one parent over here. And those alleles are going to combine so that all of the offspring are going to be heterozygous. So you can see with all heterozygous pea plants in that F1 generation, they're all going to be green. And so that would make a lot of sense then as to why this yellow trait is disappearing in that F1 generation. So 100% of the offspring will be the capital G, little g, heterozygous genotype, and 100% of the offspring are going to show the green phenotype. Remember, phenotype, physical expression. So let's try another one. If purple flowers are dominant over white flowers in pea plants, and 2 heterozygous purple flowers are crossed, what are the possible offspring combinations? So if you can, try this in your notes and then check it. You're going to put the alleles for each parent on the top and on the side. One parent on the top, one parent on the side. So we have 2 heterozygous purple flowers. That means 1 dominant allele and 1 recessive allele. And we're going to put the other one on the other side here. Again, 1 dominant, 1 recessive for the second parent. And then these are going to be the genotypes of the offspring. 1 homozygous dominant, 2 are going to be heterozygous, and 1 will be homozygous recessive, or 1 out of 4, or 25%. Will be homozygous dominant, 50% will be heterozygous, and 25% will be homozygous recessive. And because purple is dominant, 75% of the offspring will be purple. These 3 genotypes will all show purple phenotype, and then 25% will be white. So this is the way that we do a Punnett square. You're given two parents. In this case, they were the same, but you line up the genotypes, one on the top, one on the side, combine them to create the offspring, and then we list out the genotypes in their percentages and then the phenotypes in their percentages. So this is the way you'll do all of your Punnett squares. So what is the principle of independent assortment? Well, these Punnett squares that we've been doing are examples of monohybrid crosses, mono meaning one. And so these involve only one gene we've been looking at, pea pod color or flower color, just one trait. Mendel, however, watched several traits and concluded that the genes did not seem to be passed down together. This led to the principle of independent assortment. So, in other words, his data was not showing that purple flowered pea plants always had green pods, but that these genes were being passed down separately. And so this led him to predict the principle of independent assortment, that these genes were being transferred from parent to offspring separately. They were not traveling together. So in the principle of independent assortment says that genes for different traits are passed down independently from each other. So this is an example of a dihybrid cross where we're looking at two different traits, and there's no reason to freak out about this yet. We'll get to it a little bit later, but I just wanted to show you how traits can be sorted independently. So this Punnett square shows two traits, or a dihybrid cross. Pea color, which can be yellow or green, and pea texture, round or wrinkled. Notice that you find all combinations in the offspring, meaning that the genes are sorting independently. They're not green and wrinkled every time, or yellow and round, meaning that those genes are not traveling together, but they are sorting themselves independently into the reproductive cells or gametes. So what did Mendel contribute to the field of genetics? He collected extensive data on pea plants and concluded that traits were determined by genes, which were passed from parents to offspring. These genes have two or more alleles that segregate independently during the formation of gametes, or reproductive cells, in the parent individuals. So in summary, Punnett squares can be used to predict the probability for offspring to inherit a certain trait. They are based on Mendel's principle of independent assortment. Okay, so far we've been discussing complete dominance, in which the dominant allele completely overshadows the recessive allele. That's where we get the genotypes of capital B, little b, heterozygous, homozygous, recessive, and homozygous dominant. Mendel was lucky enough to have used pea plant traits because most gene patterns are much more complicated. Pea plant traits happen to be fairly simple, so he chose his specimens well. We're going to look at some more complicated methods of inheritance next. So how are traits inherited through incomplete dominance? When traits are incompletely dominant, the heterozygous phenotype is an intermediate between the two homozygous phenotypes. So here's an example. In flowers called snapdragons, the flowers can be red, white, or pink, which is of course an intermediate color. The homozygous dominant phenotype is going to be red. The homozygous recessive phenotype is white, and then the heterozygous phenotype is pink. So if we were to set up a Punnett square for this, we'd have a homozygous dominant parent and a homozygous recessive parent, creating all heterozygous offspring, and all of those heterozygous offspring would be an intermediate between the two parents. So this is looking very similar to the blending theory that Mendel was trying to fight against, but some genes do actually work a little bit like this. So this is one of the more complicated types of inheritance. And in this case, there isn't necessarily a dominant or a recessive trait. They are just combining together to create an intermediate. So there are different ways to write this. We don't always write it with capital letters and lowercase letters. But in this case, we're going to try to keep that consistent. Okay, next we're going to discuss codominance. So how are traits inherited through codominance? When traits are codominant, the heterozygous phenotype fully displays both alleles. So rather than creating an intermediate like in the incomplete dominance, this heterozygous individual is going to show both of the traits from its parent at the same time. On a side note, because neither allele is dominant, the letters are both written as capital letters. So here's our example. Some cows show codominance in their coat colors. One homozygous genotype codes for red coats, or kind of this reddish brown color. One homozygous genotype codes for white coat color, and then the heterozygous is what we call roan, which shows both red and white in the coat. And so we're using both capital letters here, and those traits are both going to be dominant, meaning they're both showing in the offspring. And so this is going to be our Punnett square for our codominant trait. And in this situation, we're going to have 100% heterozygous individuals, and they will be 100% roan, showing both traits from the parents. Multiple alleles are an even more complicated part of the genetics pattern, because many genes come in multiple forms, not just two. These genes are said to have multiple alleles. Multiple alleles can only be studied in populations because each organism still only receives two alleles, one from each parent. So they're not receiving more alleles from each parent, but there are more alleles to choose from in the population. So there's not only two alleles being passed down throughout the population. Rabbit coat color is an example of a genetics pattern that expresses multiple alleles. So some rabbits have full coat color, which would provide them with a brown fur or brown coat color. And this is represented by an allele, a capital C allele. They could also have an allele that makes them chinchilla colored. Okay, this is going to be a little c with a CH. We use a little superscript here to show which allele we're specifically talking about. Notice that this one is capitalized and this one is lowercase. So that should tell you something about the dominance and recessive traits of this genetics pattern. There can also be Himalayan colored rabbits caused by an allele that we mark as lowercase c with an H superscript. And so these rabbits are white with brown fur only on their extremities, like their feet and their ears. And then finally, there's also albino rabbits in this population, and they have simply the recessive allele, which makes them fully white. So each of these alleles is dominant over the next. And this can be determined by following patterns of inheritance through the populations. So we now know that full color alleles are dominant over chinchilla colored. Chinchilla colored alleles are dominant over Himalayan colored, and Himalayan colored alleles are dominant over albino. So only when a rabbit would inherit two little c's, a little c genotype, would they actually show the albino phenotype. If they had, let's say, a chinchilla-colored allele and an albino allele, they would show the chinchilla-colored trait and be gray. So each rabbit has a combination of two of these alleles, but you can see that there are multiple in that population. Polygenic traits are just as you might seem from the word polygenic. Poly means many, genic meaning genes. So these are traits that are controlled by two or more genes. Even with the multiple alleles, we were still talking about one gene for rabbit coat color. It just happened to have multiple versions of it. Now we're talking about more than one gene. These genes may be on the same or different chromosomes, and they may have two or more alleles. So we're getting very complicated here in our inheritance patterns. As you might expect, this provides a wide range of phenotypes. And most human traits are polygenic, including skin color and eye color. So skin color is produced by melanin, a pigment in your skin, and there are many genes that code for the amount of melanin that is produced in the skin. And so that gives a wide range of different phenotypes of skin colors. Are there any other factors involved in gene expression? Of course there are. The internal and external environments of an organism can also affect how traits are expressed, just to make it more complicated. Internal factors like hormones and immune response can control whether genes are actually displayed in a phenotype. And external factors like lifestyle, smoking, lifting weights, exposure to sun and temperature can also cause some genes to be expressed differently. So in a human population, if we discuss lifting weights, let's say you had two parents that were very thin and had small frames, you would expect that the child that they have would be also have a small frame, maybe be skinny or thin. But lifting weights is an external factor that can change the expression of that trait. Sea turtles also do this. They lay their eggs on sandy beaches, and the temperature of each egg is able to change the gender. So they are changing the expression of the genetic traits in that egg simply by adjusting external environment. So in summary, incomplete dominance, codominance, and multiple alleles are all exceptions to Mendel's rules. Some genes work together to create traits, and some are affected by the environment. Okay, so we have discussed how genes cause traits. But in this section, you'll learn exactly how those genes get passed from one generation to the next. Obviously, we've discussed that genes do get passed from one generation to the next, and that they get passed in a dominant recessive alleles, and that they have independent assortment, but now we're going to discuss what is that process of actually moving the genes from one generation to another. Hint, it's a new type of cell division called meiosis. So as we get into the study here of meiosis, we're going to get through some terminology first. Then we'll go through the overview of the process. So let's start with haploid and diploid cells. How do haploid and diploid cells differ? Well, most cells in an adult organism's body contain two sets of chromosomes. One set of chromosomes came from their mother, and one set of chromosomes came from their father. Only the reproductive cells have only one set of chromosomes. So this is a picture of a muscle cell here. In all of your muscle cells, and all of your eye cells, and all of your stomach cells, and all of your skin cells, you have two sets of chromosomes, and they're all the same. And those are called somatic or body cells, and we call those two sets of chromosomes, the cells that contain those two sets, diploid cells, di meaning two. Only in your reproductive cells, or your gametes, either sperm or eggs, do you have one set of chromosomes, and those cells are known as haploid. A good way to remember this is haploid meaning half of the typical number of chromosomes. So let's take a look at what these chromosomes might look like, and then we'll get into the process of meiosis. So in a diploid cell, the pairs of chromosomes are called homologous chromosomes. Remember, homo means same, so these are similar or matching chromosomes. Homologous chromosomes have matching genes. One chromosome came from the mother and one came from the father. So if these are homologous chromosomes here, they're going to have the same arrangement of genes. So maybe this gene segment up here codes for skin color, and this gene segment down here codes for hair color. But they are going to have the same set of genes on both of those homologous chromosomes. They may have different alleles on those two chromosomes. So this hair color gene might be a dominant allele from the dad and a recessive allele from the mom, but the genes are matching. When those chromosomes duplicate during cell division, they form a set of 4. So we've got 2 chromosomes here. When they replicate, you have 2 sister chromatids, just like in mitosis, and 2 sister chromatids for this chromosome. And because there are 4 of them, we call them a tetrad, tetra meaning 4. Okay, now that we've gotten that terminology out of the way, what is the real purpose here? Why is there a different cell division process for sex cells than there is for any of the other cells in your body? Well, in order to have haploid gametes, or reproductive cells, the cell division process must be different than that of somatic cells. So as a review, what is the cell division process called in somatic or body cells? It's called mitosis. And again, as a reminder, in mitosis, the goal is to produce identical cells from the cells that we started with. So when you're producing more skin cells through the process of mitosis, those skin cells are all going to be identical to each other and identical to the parents. But the process of meiosis creates haploid gametes. So the goal here is to create half as many chromosomes at the end of this process as we started with. And so that has to be a different process than mitosis. So why is it important for gametes to be haploid or have half the usual number of chromosomes? Think about this for a second. Well, the gametes, or sex cells, are going to join together during sexual reproduction. If the chromosome number wasn't reduced when we formed these cells, the resulting zygote would have double the correct number of chromosomes. And then in every generation after that, it would continue to double, which is going to completely change the genetics of that population. And that number of additional chromosomes is not going to allow an organism to survive. So this is definitely not what we want. So in order to reduce that chromosome number, meiosis is going to begin the same way as mitosis, with chromosomes duplicating. We just saw that in the tetrads. The chromosomes are going to duplicate and be joined at the centromere here. And they are going to line up and separate in meiosis 1, which is the first half of this process. There is going to be another period of cell division. So unlike mitosis, there are 2 periods of cell division, and that reduces the chromosome number by half. So let's get into the details here. As the parent cell is preparing to divide for the first time, which we call prophase 1, the homologous chromosomes in a tetrad can trade genes in a process called crossing over. So here's what this looks like. As these chromosomes have duplicated, their arms can actually extend over each other and switch genes. The DNA breaks off and switches from one chromosome to another. Crossing over is why siblings have the same parent's chromosomes, but a different combination of genes. It makes each daughter cell slightly different. So if we go back and look at our full process of meiosis here, you can see that in prophase 1, these arms are crossing over and exchanging genes, and they are separating in that first half of meiosis. And then in meiosis 2, those chromatids are separating, and then you end up with 4 different cells that have a different combination of genes. And this is, of course, with just 2 chromosomes, which you have 46 chromosomes, so there's going to be an even greater amount of gene exchange, so that these are going to be completely different cells by the end. So in summary, while most of the body's cells are diploid, gametes are haploid, so they can join during sexual reproduction. Meiosis creates these haploid cells and begins with pairs of homologous chromosomes. So just as in mitosis, there are phases of meiosis that we discuss throughout the process. In meiosis 1, the first half of the cell division process, the homologous chromosomes cross over and then separate. Meiosis 2 is just like mitosis. So in meiosis 1, we have 4 different phases. Prophase 1, where homologous chromosomes pair up into tetrads and then cross over. Metaphase 1, where homologous chromosomes line up into pairs along the middle of the cell. Anaphase 1, where homologous chromosomes separate. And telophase 1, where homologous chromosomes reach the ends of the cell. So this is sort of like mitosis, but it's a double form of that because we're working with sets of chromosomes. So here's what that looks like. In prophase 1, homologous chromosomes are pairing up into tetrads and crossing over, exchanging genetic information. In metaphase 1, those homologous chromosome pairs, those tetrads, are lining up in the middle of the cell. In anaphase 1, homologous chromosomes separate, so whole duplicated chromosomes are being pulled to the ends of the cells here in anaphase 1. And then in telophase 1, we've now successfully separated the tetrads into their individual chromosomes, but they're still duplicated chromosomes. In meiosis 2, which is going to be just like mitosis, we now have these duplicated chromosomes, and we're going to separate those chromosomes, separating the sister chromatids on each half of that replicated chromosome. So in prophase 2, the nuclear envelope disappears. In metaphase 2, chromosomes line up in the middle of the cell. In anaphase 2, the sister chromatids are going to separate, so that centromere breaks and the sister chromatids pull apart. And in telophase 2, sister chromatids reach the end of the cell and the nuclear envelope reappears. So just like mitosis here. But again, we're going to end up with different cells at the end of this process than we started with. So how do mitosis and meiosis differ? Well, first of all, in mitosis, if we start with one parent cell and one parent cell in meiosis, we will come up with a different number of daughter cells for these two processes. So meiosis starts with one parent cell and ends with 2 daughter cells. It only has one process of cell division. The cell divides one time. Meiosis begins with one parent cell and ends with 4 daughter cells because the cells go through 2 periods of division. Are the daughter cells identical to the parent cell? In mitosis, yes, they are. In meiosis, no, they are not. Because of that genetic exchange of information during crossing over, all of those meiosis gametes at the end, those daughter cells, are going to be different from each other and also different from the parent, because otherwise you would look identical to your parents. So that's obviously not happening in the process of meiosis. The purpose of the process here is different. Mitosis is for healing, growth, and repair, replacing dead cells. Meiosis only has one purpose, and that is for creating gametes or reproductive cells. We call the process of mitosis an asexual reproduction process because it doesn't require 2 individuals, but the meiosis process is ultimately going to involve the combination of that gamete with another individual. So that's a sexual reproductive process. In summary, the process of meiosis involves 2 cell divisions, resulting in 4 daughter cells. Because of crossing over, the daughter cells are all different from each other and different from the parent cell. So now that we've looked at reproductive cells and how these cells are formed, we're going to take a look at one more pattern of inheritance, which is called sex-linked traits. So this is another way that genes are passed down, but it depends on the different reproductive cells. So it's passed differently in sperm and eggs. So now that we've discussed reproductive cells, let's talk about this inheritance pattern. So how are sex-linked traits inherited? In humans and other mammals, biological sex is determined by a pair of inherited sex chromosomes, known as the X and Y chromosomes. So a genotype of XX, or 2 X's, one from each parent, creates a female, and XY creates a male phenotype. During fertilization, which is the joining of eggs and sperm, an egg cell and a sperm cell, the egg contributes an X because all females are contributing X chromosomes in their egg cells. The sperm may contribute an X or a Y because it has both. And keep in mind as we discuss this that this is a very simplified view of biological sex. Even when we're discussing biological sex and not gender, there can be some variations, but this is the simplest way to discuss it. So the egg cell is donating X, and the sperm cell is donating X and Y, and these are going to segregate independently just like other genes can. And then we'll have half offspring that have XX genotypes and half offspring that have XY genotypes. Although these chromosomes determine sexual development, they also contain genes for other traits. So they don't just determine biological sex. They also have genes for a variety of other traits. Because those genes are found on the sex chromosomes, though, they are called sex-linked traits. So they are linked to the sex chromosomes, and so they travel based on sex chromosomes. Examples of these traits include red-green color blindness, male pattern baldness, and hemophilia, which is a blood clotting disorder. So in order for a female to show a sex-linked trait, both X chromosomes must be affected. A male only needs to inherit one affected X. So here are the phenotypes for sex-linked traits. 2 normal X chromosomes, or chromosomes that don't contain the gene that we're discussing, will create an unaffected female. So a female that does not show that trait, does not have that disease, whatever we're discussing. If one of the X chromosomes in a female contains an allele for that trait, she will be called a carrier if the other one does not. So if it's only one X chromosome that's affected, she will be a carrier female. If she has 2 X chromosomes that are affected, she will show the trait. She will be affected by the trait or affected by the disease. Males only have 2 different phenotypes. They can either be XY, meaning that they are unaffected because there's not an affected X here, doesn't show that trait. If this one X is affected, they don't have another X to protect them like a carrier female does. So they will show the trait and be affected automatically with just that one affected X. So we'll do a Punnett square to show how this works. These are interesting traits, but they are a little bit trickier than the other traits we've been working with. So that's why we saved those for the end. So let's try this cross for 2 individuals with alleles for hemophilia. Let's say we have a carrier female who inherited one X chromosome that happens to have that hemophilia gene on it, or hemophilia allele. And if she reproduces with a male that also carries that hemophilia trait, we'll do a Punnett square to show how this will turn out for their offspring. So their offspring could have the possibility of being female with the trait, with hemophilia. They could be carrier female. They could be a male affected by hemophilia, or they could be an unaffected male. So we write these out as separate percentages. 25% XH, XH, H for hemophilia, and 25% XHY. These 2 quadrants of our Punnett square are both going to be individuals that have the hemophilia disease. Then we also have 25% that are carrier females and 25% that are unaffected males. Neither of these quadrants, these offspring, are going to show the disease for hemophilia. This individual here, this genotype, is going to be able to pass down that gene to her offspring, but she's not going to show the disease in her lifetime. So that's how we do a sex-linked Punnett square. Now finishing off this unit here with pedigrees. A pedigree is a graphic representation of an individual's family tree, which allows scientists to see inheritance patterns. So a pedigree is simply a model that's used to show how a lot of different predictions through generations can line up. So you can either look back through generations in a pedigree or make predictions about the future using Punnett squares. And there are specific symbols that we use to discuss a model of a pedigree. We use a blank or empty square for unaffected males. We use a blank or empty circle for unaffected females, a filled-in square for an affected male, a filled-in circle for an affected female, and a half filled-in circle for a carrier female. So here's an example of a pedigree for a sex-linked trait. Practice drawing a pedigree of your own. So here in this pedigree, we have one carrier female, and this pedigree line here shows that these two individuals reproduce. We usually say they're married, but they may not be. So they've now reproduced, and their offspring are represented by this vertical line here, and we put all of the offspring in the next generation. So these two individuals, a carrier female and an affected male for whatever trait we're talking about in this hypothetical pedigree, they had 4 offspring. One is a carrier female, one is an affected female, this one is also an affected female, and then an affected male. And this carrier female married or reproduced with an unaffected male. And then we can see the results of their offspring as well. And so you can see how a trait can be passed down from one generation to the next and follow the pattern of inheritance. So this can be used to determine the genotypes of parents based on their offspring if we don't know the genotypes of the parents, or it can be used to show predictions for the genotypes of offspring if we do know the parents. So a pedigree is a really helpful tool for following predictions of Punnett squares and how traits are passed down from one generation to the next. So in summary, sex-linked traits are carried on the X and Y chromosomes. These traits affect males and females differently, so it can be helpful to show their inheritance in a visual pedigree.